Hi, everyone. I'm going to give it a couple minutes to see how many people join. I'm going to give it one more minute and then we can begin. Okay, so welcome everyone. We're glad you can join us today for the NIH Office of Dietary Supplements seminar discussing dietary supplements and chronic disease. I'm Jasmine Hayes, and I'm a health scientist administrator here. And in addition to organizing the seminars, I also lead the co-funding program. And before we welcome today's speaker, let me take a moment to go over a few brief announcements. I'd like to remind you that our next ODS seminar is coming up. This seminar will discuss nutrigenomics and dietary supplements. The seminar will be held Wednesday, January 21st. If you're not already on our seminars mailing list and would like to be, please check the chat. We'll share an email address there that you can request being added to the ODS webinars mailing list. A quick note about webinar logistics. To ensure good quality audio, everyone will be muted, and we ask you to remain muted until the Q&A. If you experience any technical issues with audio, video, closed captions during the webinar, please use the chat, and one of us will be happy to assist. And as a reminder, please be considerate to everyone so we can all enjoy and learn from today's speakers. Okay, and now I have the pleasure of introducing today's speaker. Dr. Howard Sesso is an associate epidemiologist at the Division of Preventive Medicine at Brigham and Women's Hospital, associate professor of medicine at Harvard Medical School, and associate professor of epidemiology at the Harvard T.H. Chan School of Public Health. He designs, conducts, randomizes clinical trials and epidemiologic studies focusing on dietary supplements, nutrition, and lifestyle factors to prevent cardiovascular disease, hypertension, cancer, and other aging outcomes. Dr. Sesso is an associate director of the Division of Preventive Medicine and director of nutrition and supplement research at the Osher Center for Integrative Health. Dr. Sesso has been involved in several medical trials, Division of Preventive Medicine, helping lead the Physician's Health Study, or PHS, conducting several ancillary studies, vitamin D, omega-3 trial, or VITAL, and co-leading the cocoa supplement and multivitamin outcome study. He has also conducted several small-scale clinical trials, testing supplements and nutrition, plus initiated an epigenetics working groups stemming from several research grants across PHS and vital in Cosmos. So we are going to enjoy Dr. Sesso's presentation. He's going to be discussing how hybrid clinical trials of dietary supplements delve deeper into mechanism and efficacy. And Dr. Sesso, the microphone is yours.
Thank you so much, and hopefully everyone can see the screen here. It's a pleasure to be here, pleasure to be with all of you. I'm also happy to see that the seminars are restarting now with things moving forward. I'm going to be offering my perspectives, looking at the Cosmos trial and also a few other the trials that we've been involved in, looking at how we can design better trials to understand more about the effects of dietary supplements, particularly with regard to mechanism and efficacy. Just some disclosures. The work that I've been involved in for more than 2 decades now really can't be done without industry support and collaboration. This includes Haleon with regard to Cosmos and the Physicians Health Study 2, as well as Mars Edge for Cosmos in particular. And I've got other disclosures for other projects that I've been undertaking. So I want to start with a few general concepts when we think about chronic disease, dietary supplements. Chronic disease, I think, sometimes kind of misguides us a little bit about thinking about prevention or treatment or management of things as we're older, when in fact, it's really more about the life course and healthy aging. And it's also more, obviously, part of it is increasing life expectancy, but a lot of it really revolves around the idea of looking beyond the clinical outcomes and getting into brain health and wellness outcomes and improving quality of life. And again, this isn't true not just when we're middle-aged and older adults, but it starts at birth. It starts even before birth. So we need to be thinking about dietary supplements in clinical trials, particularly hybrid clinical trials across the age spectrum in its complete form. I think the other point that's also worth noting, and we already know this, is that diet is incredibly complicated. This is just one example of a schematic. When we think about, maybe it's maybe it's parts of the body here on the left side, or some of the specific essential vitamins and minerals and trace minerals and the food sources. How do dietary supplements fit in? Well, I think sometimes as much as we think diet is complicated, I think it's very fair to say that dietary supplements are just as complicated. And this is not necessarily a function of the DeShays Act, the 1994 DeShays Act that had come forth that, of course, established the Office of Dietary Supplements now more than 3 decades ago. The original charge, among others, was to think about promoting the scientific study of supplements for maintaining health and preventing chronic disease. We know it goes well beyond that now, today. And even how we define dietary supplements, of course, continues to evolve. It's not just a dietary ingredient taken by mouth in some form. It's not just the essential vitamins and minerals, but we have a host of other types of compounds and botanicals that are included here. And they come in lots of different forms. And of course, not just DeShay, but as we look at our regulatory framework and thinking about labeling of products for dietary supplements, it's a very complicated picture. And just as one example of that, much of my work over the years, and I would say most of the presentation that you'll hear here, revolves around multivitamin, multimineral formulations. Now, these remain the most commonly used dietary supplement in the U.S. among all adults. But it's a very interesting and complicated story. It's not a simple one-size-fits-all when you think about how even multivitamin, multimineral supplements can differ on lots of levels. It's obviously going to differ on formulation. There are just sometimes more subtle, sometimes more not so subtle differences in combinations and amounts of vitamins and minerals, other bioactives or herbals or other compounds that are added in there. Target populations. This is a more recent evolution. It kind of used to be a single type of multivitamin that everyone would consider taking, but now it's become very specialized for kids, for men, for women, for older adults. And also thinking about specific outcomes in mind. It might be bone health or eye health or brain health. And anyone going to the store shelf today, and this is just one picture as an example, is really overwhelmed, not just for specific individual vitamins and minerals and bioactives, but especially even in the multivitamin space itself has its own complexity to it. So how do we disentangle it? Well, it's not easy when we do scientific studies, we tend to focus on individual dietary supplements, examples of that. A lot of the work that I'll be showing you today from Cosmos is really derived from the Physicians Health Study 2 trial. This was a part of a 2 by 2 by 2 by 2 factorial trial that included a multivitamin at the time in middle-aged and older men. And when we had published these results now more than 10 years ago, there were some interesting findings that served as the foundation in part for the Cosmos trial. One is that we had seen that those men that were randomized to take a multivitamin daily had a small but statistically significant 8% reduction in total cancer. And we saw for other site-specific cancers a fairly consistent lowering of risk, whether significant or not. More interestingly, we saw that for the, men that were age 70 and up, the effect actually strengthened to an 18% reduction, although that interaction, that statistical interaction by age did not differ compared to younger men. We also saw that those men taking a multivitamin actually had a lower risk of developing cataract, 9% lower risk, as well as having cataract surgery. So there were some pretty clear signals that had emerged from the Physicians Health Study 2 trial that helped in part to initiate the cocoa supplement and multivitamin outcome study, or COSMOS. And as you'll see, this is an example of what we would consider to be a hybrid clinical trial testing a dietary supplement. That is a framework that we want to think more carefully about as we consider other clinical trials moving forward. Now, one thing I want to acknowledge is that I'm one of two multiple PIs of the Cosmos trial. My other is Dr. Joanne Manson. It was really with her effort, along with many others, as you'll see, that Cosmos not only came to fruition, but also has flourished over the last several years, in particular with recent publications that I'll be highlighting. Cosmos more broadly is consisting of more than 21,000 older women and men across the US. So women age 65 and up, men age 60 and up, that were initially free of CVD and also recently diagnosed cancer. When we look at Cosmos, it's a 2 by 2 factorial trial. I'll talk a little bit about the cocoflavanol component in a bit, but we'll be emphasizing a little bit more on the multivitamin components of the Cosmos trial findings. Median follow-up was 3.5 years. Our primary outcomes were major cardiovascular events as well as total cancer. And as you'll see, we've been leveraging our hybrid design with bloods and clinic assessments to further enrich our ability to look at mechanism and efficacy in different ways. Now, one other point worth noting, now this is a deliberately complicated slide, but when we look at dietary supplements and doing clinical trials, whether large or short or small, collaboration is key. We have seen in our experience and knowing others in the field over the years that these types of trials cannot be done, especially with dietary supplements, without collaboration across industry partners, as well as foundations and consumer health groups, of course, the National Institutes of Health and the various institutes that are involved, including the Office of Dietary Supplements, and then we here in the hospitals and the academic institutions as well. All of this collectively is what transforms the science into more definitive science, and especially through randomized clinical trials. And as you'll see, Cosmos has been very much a successful product of that, and also hopefully a framework moving forward for how we can better conceive new trials in different areas looking at dietary supplements across the life course. I do want to comment briefly on the cocoa extract intervention of Cosmos. There was, leading into Cosmos, a fair amount of evidence actually on some of the mechanisms of effect, but not necessarily a trial that was looking at clinical cardiovascular endpoints in Cosmos. And this was one of the main reasons that the Cosmos trial was conceived. I will not be showing these data, but I have these highlighted in green. We've had some recent, oops, my apologies. We've had some recent papers that have come out that have highlighted some of the cocoa extract's potential benefits with hypertension in particular, as well as C-reactive protein. But more to the point, when we looked at the main findings for the cocoflavanol or cocoa extract intervention in Cosmos, we saw some interesting findings both for intention to treat analyses as well as per protocol analyses that raise some other broader questions about how we look at the trial data and how we analyze it, particularly when we're looking at diet, nutrition, including dietary supplements. Overall for Cosmos, for our primary endpoint of total cardiovascular disease, which encompassed many of the, actually all the secondary endpoints you see here, except for all-cause mortality, we saw a 10% reduction, although it was not statistically significant. Interestingly, when we started to look at each of the individual endpoints for cardiovascular disease, as well as other combined endpoints, we saw a consistent lowering of cardiovascular risk. And in particular, we saw that for cardiovascular disease death, there was a statistically significant 27% reduction for those taking cocoa extract daily versus placebo. Furthermore, under other outcomes, this was not pre-specified, but if you were to look at a typical MACE outcome, we saw a significant 16% reduction for cocoflavanols on MI, stroke, and CVD death. But what's interesting is that when we compare the intention to treat analyses with the per protocol analyses for those who were compliant with the intervention, taking 3/4 of the pills on average daily, we saw that the effects actually strengthened. Now, of course, this breaks the beauty of randomization in clinical trials, but it does highlight some of the important considerations when we look at trials of dietary supplements and understanding how the compliance-based analyses might give us a little bit more information and context, understanding that those who are actually taking the cocoa extract pills in this case actually had stronger effects on the cardiovascular endpoints of interest. Furthermore, in Cosmos, and we'll be discussing this a little bit for the multivitamin intervention, having an objective biomarker of not just compliance, but of intake is essential. In the case of cocoflavanols, we have gamma valerolactone metabolites, or GVLM, which is a validated marker of dietary flavanol intake. What we had in Cosmos is a subset of more than 2,000 participants who had provided baseline and at least one spot urine sample during follow-up from which we can measure GVLM levels. And sure enough, as you can see, in red is the intervention group, blue is placebo. Those that were taking the cocoflavanols actually had a more than threefold increase in GVLM. And this serves as an important point for these nutritional trials that we do in dietary supplements of having these objective biomarkers. And finally, there's been increasing momentum thinking not just beyond, thinking really beyond the vitamins and minerals and thinking about guidelines, dietary bioactive guidelines, considering things like, in this case, flavin-3-ols, not just from the findings from the Cosmos trial, but other trials and other work that has been done. And these are areas that I think ought to be considered moving forward. So I'd like to turn our attention now to the multivitamin intervention. For Cosmos, our main finding, we were not able to replicate exactly what we'd seen in the Physicians' Health Study 2, which was of male physicians. In Cosmos, which included both men and women, we saw that for our primary endpoint of invasive cancer, the hazard ratio was 0.97. There was no overall effect of a multivitamin to lower the risk of invasive cancer. Although it gets interesting, in the Physicians' Health Study 2, you see this in this kind of purple box. We see that the results for men were actually a hazard ratio of 0.94 versus 0.92 in PHS2. Lung cancer, we actually saw a significant 38% reduction in lung cancer, and then all-cause mortality doesn't seem like a huge effect here at 7%, but you compare it with the Physicians' Health Study 2 at 6%, we start to see some conversions here that gets interesting. It should also be noted we did look at cardiovascular events. We saw no effect of a multivitamin on total cardiovascular disease. Where I think it gets interesting is that the U.S. Preventive Services Task Force had updated their recommendations for multivitamin supplementation after the Cosmos trial results were published. And while their overall recommendation was that the evidence was still insufficient, one can start to look at some of the summary data and make an argument to say, well, yes, maybe it's insufficient, but maybe there is something here, even as a modest effect, that might be quite important. And as two cases in point, if we look at first any cancer, combining the other trials that have been done, there was a small but statistically significant 7% reduction on any cancer for those taking a daily multivitamin versus placebo. Now, 7% doesn't sound like a lot, but when you think about it from a public health standpoint, that could actually be quite meaningful. Similarly, for total mortality, for Cosmos and PHS2, we saw like a 6-7% reduction. This was not statistically significant when you put all the data together when it was meta-analyzed. With additional trials, we might look at this a little bit different. Again, it's not a very large effect, but it's a fairly low-cost intervention. And I think it's a question that ought to be reconsidered moving forward as we have more data to hopefully support it. But the beauty of Cosmos, as well as I would say the VITAL trial, which I believe many of you are familiar with, is that we have at the Division of Preventive Medicine adopted what we call this hybrid randomized clinical trial design. And on one hand, we design these trials, of course, with primary clinical outcomes in mind, but this can realistically extend into the healthy aging space, and not just in older adults, but even in younger adults. And the idea is that when you're conducting a trial, whether it be large or small, short or long, that beyond the primary outcome, it's imperative to develop ways to collect either biospecimens or other types of assessments that can be built in addition to what the trial is primarily designed for, to be able to take advantage of the hybrid design to look at mechanisms of effect, as well as on the left side, thinking about effect modification, who's more likely to benefit or not. And this starts to get into the issue of not just precision nutrition, but the role of baseline nutritional status, which is usually pretty important when we think about most dietary supplements. So it's this design that you'll see from Cosmos thus far that has really helped to expand the scientific landscape and offerings and understandings of how we'll be discussing the multivitamin, but also the cocoa extract intervention could be coming into play here with different health outcomes. Now, Cosmos has benefited not just from some of these ancillary studies that are listed, but others that have developed over time. We made a conscious effort through a combination of support through either NIH and or support from Marsden Bioscience at the time, but also from other projects that you'll see on the next slide. Incorporating additional data really helps to magnify the utility of the trial, the scientific reach of the trial. And we've seen this not just with Cosmos, but also with our group's VITAL trial as well. And in fact, what comes of this, it really turns into a much more expansive data matrix. And I have here just around the core data that we've collected in Cosmos, just a sampling of some of the other projects that have emerged, other data sets, other funding that has emerged from this. And this comes back to that other slide that I had presented that had showed all the different ways that collaborations are key for dietary supplement trials. I think Cosmos in many ways has been a nice example of this, and one that we should think about replicating moving forward in different ways. I think the other thing worth mentioning is that, when we think about trials of dietary supplements, especially the hybrid design, it also does a much better job thinking about some of the other core concepts through the NIH. As one example, thinking about the whole person health initiative that's coming through NCCIH. When we're thinking about mechanistic pathways, signaling pathways, yes, we want to think about the whole person, of course, but understanding its connectivity of a multivitamin or a cocoa extract supplement with regard to organ and systems-based research become other ways that we can think more carefully about how the trials can help feed into these types of networks. And furthermore, of course, trials such as Cosmos and others in the dietary supplement space fit very well into the strategic plan for nutrition research at NIH. Precision nutrition, however you want to characterize it, ultimately requires accurate and reliable dietary assessments. Objective nutritional biomarkers, as you've seen already from GVLM for the cocoa extract intervention, we have that available for the multivitamin intervention. And thinking about things like multi-omics. Those highlighted in purple, I'll be presenting at least some data on just to provide some context. But in a way, dietary supplements are a unique research opportunity that fits well into these plans, because dietary supplements, for better or for worse, are much better positioned for precision nutrition than food-based interventions. It's not that I'm against food-based interventions and dietary patterns. Of course I am. But because of placebo control, dietary supplements are in a much stronger position. It's easier to implement, easier to test, easier to draw those conclusions more directly. Again, diet is complicated. Supplements are complicated too. And when we looked in the Cosmos trial at the effect of the multivitamin intervention on nutritional biomarkers, for example, not surprisingly, if it's in the multivitamin, you'll see those levels go up. You'd be surprised in longer term trials that have been done in dietary supplements, not just Cosmos. Many of these trials have not necessarily had, I would say, the luxury of having follow-up biospecimens. So it's reassuring to show that if it's in the multivitamin, it goes up. Vitamin D, 25-hydroxy-D, vitamin B12, folate are three examples. We have other work that is now under review, looking also at different carotenoids and also alpha tocopherol measured in Cosmos at multiple time points over time. And again, not surprisingly, if it's in the multivitamin, it went up. The microbiome is also an area of interest for us in Cosmos. We actually had collected just over 300 samples from participants, both at baseline and then again at 2 years follow-up. And we've done some pilot work. This is just specific to the multivitamin intervention, but we've also seen this for the cocoa extract intervention. And we've seen some preliminary evidence that with the dietary intervention, there are changes in the microbiome. Now, one, of course, key question when thinking about the microbiome in the context of dietary supplements is that, first of all, what do the changes mean, and how do those changes factor in to or mediate the effects on different disease or other clinical outcomes that might be of interest? And we'll see this come into play, not with the microbiome, but with some other work that I'll show you. Another area of interest is biological aging. Biological aging, of course, is a hot button topic right now, and in good ways. I mean, we think about aging typically in terms of chronological age, shown up in the upper right corner here. As we get older, time marches on. It's standard. It's exactly 1 year to 1 year. But we also now appreciate that there's mounting evidence that shows how biological aging can accurately and rapidly predict not just functional status, or sometimes is deemed intrinsic capacity, but it's not just getting older, but it's also how we are aging too. Now, at the bottom, there are many different, actually on the right side here for this figure with the circles, and as well as on the bottom, there are many different ways that have been cited that could be biological age estimators. They could be visible biomarkers that are measured, excuse me, sorry. Visible aging biomarkers, they could be health and disease markers, blood pressure, glucose, A1C. You can start to look at it on a molecular or cellular level, and of course, omics. And I've circled these three for omics. I've shown you briefly the microbiome, but we'll be going into a little bit more detail on epigenomics and also metabolomics. And just to expand on epigenetics a little bit, there are really 3 generations of clocks, and there's much more that go beyond this that help to predict various forms of biological aging. And again, for those that are unaware, when we think of epigenetic clocks, these are derived from DNA methylation features at the various CPG sites on DNA that can help to regulate gene expression. And this is independent of changes in the genetic sequence itself. We use algorithms that have designed or developed these epigenetic clocks, our so-called first generation clocks are the Horvath and Hannum clocks. Second generation are focused more on mortality as a predictor, Grimm age, pheno age, and then the pace of aging, which is the Dunedin pace clocks as well. And the idea is people might be having accelerated or decelerated aging. I might be 50 years old, but if my biological age is 45, well, I'm clearly not aging as poorly as I think I might be. On the other hand, if my chronological age is 50 and my biological age is 55, what's going on there? Are there health outcomes, other explanations? So in Cosmos, and this was through support in part from the National Institutes, the NHLBI, we've conducted an epigenetic study. And the aim here was to look at the 2-year effect of the multivitamin supplements as well as the cocoa extract supplement on biological aging through epigenetics. Again, hybrid trials. We have the overall trial, 21,000+ participants. We had almost 7,000 people provide bloods at baseline. We were able to tap into nearly 1,000 people who had bloods collected at baseline 1 and 2 years, and we're able to measure DNA methylation and generate some of the clock data that I'll be presenting to you here. Now, first thing that's worth noting is that longitudinal changes in clocks just over time in otherwise healthy participants, of course, we live to be, 70, 80, 90 years old, hopefully. But at the same time, 2-year changes are not great in a normative population. This is work from Dr. Rick Hamaya, where if you just look at normal changes in these clocks over 2 years, the changes aren't great. So the key question is, what are the types of interventions that can actually move or change the trajectories or change these clocks and how they're progressing over time? Another way of looking at it is, even if you look at the changes in the clocks themselves and how they correlate with each other, I think the key thing to note here is on the left side that these correlations between the clocks at different time points and the changes are quite modest. So the key is, what interventions, whether it be through diet, lifestyle, pharmacologic, can really move or change the trajectories of biological aging. And when we think about dietary supplements and changes in epigenetic clocks, of course, diet is paramount, right? We want to start with diet, but we're also appreciating the fact that we can look carefully at the effect of dietary supplements on long-term morbidity and mortality, but yet looking at the changes in the clocks provides another layer of intrigue and perspective mechanistically. And we can look both at the changes in epigenetic clocks, looking at it as a modifier of the outcome, so effect modifier, as well as a predictor of outcome. I'll be showing you data from Cosmos. We have work, underway in the VITAL trial, and then we have actually a quite large resource within the Physicians Health Study 2 and 1 trials to look at it as an effect modifier in particular. So these are analyses that are looking at the effect of the daily multivitamin in Cosmos on yearly changes in the epigenetic clocks. Again, clinical trials, the beauty of these is that randomization tends to work. Mean age in Cosmos is just over 70 years in this particular sub-study. And through the beauty of randomization in trials, we don't really worry about confounding because these are distributed in a way that makes sense. Doesn't take away from the questions of generalizability, of course. Our main finding that we saw in Cosmos, and this work is, I think, forthcoming publication actually, is that particularly for the mortality-based clocks, PC Grim age and PC Pheno age, the third and fourth lines that you see here in purple, we saw that those that were on a multivitamin had significant improvements in biological aging compared to placebo for each of these clocks. The other clocks that we had tested also trended the same way, but just we did not reach statistical significance. Now, there's a lot on this plot, admittedly, but I think the key finding to draw from this is that for most risk factors that we might consider for healthy aging or different clinical outcomes, so whether you're obese or if you're hypertensive or had diabetes, the effect of the multivitamin intervention was better on these epigenetic changes, particularly for the mortality-based clocks, compared to placebo. In particular, I want to highlight what I've circled here in these red dotted lines, that it's also a function of where you start. So if you start in an accelerated biological age, in other words, I might be 50, but my biological age is 55, actually I benefited much greater from taking a multivitamin compared to those that were on placebo. And those that were healthier, shall we say, in terms of biological age at baseline, did not benefit as much from taking a multivitamin. Interesting finding that we're looking to expand upon as well. Now, one of the nice things of Cosmos and the purpose of these hybrid trials is that we can start to combine the different projects together. So as one case in point, you can ask the question, why are there larger benefits among those who are of greater biological age in Cosmos? So we can take a cross, if you will, of those who were in the epigenetics study as well as those who provided nutritional biomarkers. And we can see that, and without going through all the details of this, when we look at some of the carotenoids, folate, vitamin B12, and 25-hydroxy-D, that first of all, lower levels of nutritional biomarkers were linked with accelerated biological aging at baseline. Good to know. Nutrition matters. But more importantly, there were larger improvements in nutritional biomarkers, and those were inversely associated with epigenetic aging during follow-up. So this provides at least one insight as to why there might be improved biological aging on those taking a multivitamin. It might be through the improvements in these more objective nutritional biomarkers. Now, piggybacking in part with this is also a metabolomics study. This was a little bit smaller in scale compared to the epigenetic project that I just showed you. This is another project that's also currently under review with the journal. Here we took 400 participants in Cosmos that provided bloods at baseline 1 and 2 years. We used the Nightingale panel for NMR metabolomics profiling, and not just looking at the individual metabolites, but also now there are metabolomic clocks of biological aging that have been generated that have been trained either on chronological age or on all-cause mortality. And they've also been linked with various metabolomic risk scores for different chronic diseases. So again, when you're leveraging these studies in a hybrid trial design, this is a trial. It's a trial and design. There is no confounding in theory. Always want to check anyway, but the nice part about this is that randomization will continue to work. The main finding to report here is that when we look at the effect of a daily multivitamin on some of these metabolomic risk scores, we find that in a way, this is a bit more hypothesis generating, first of all, right? So we're looking at changes in the individual metabolites. And I did not show you those specific data, but when you put the various metabolites that are coming off of the panel and converting them into these risk scores, we see some pretty interesting findings that emerge here comparing the multivitamin versus placebo groups. First of all, one's a little bit contradictory. Oops. One is showing that for major cardiovascular events that there seems to be improvements for being on a multivitamin versus placebo. As I showed you for Cosmos as well as for the Physicians Health Study 2 trial, we did not see that type of an effect for clinical outcomes that we actually captured. On the other hand, when we look at something like all-cause dementias, you'll see when we looked at cognition in the Cosmos trial, we do see some interesting findings, as I'll show you. The cataract finding here that we had seen at least for the Physicians Health Study 2 was also corroborated here, looking at the metabolomic risk score. It also extended to some of the cancer sites, which is interesting. Lung cancer did not emerge. One thing that was interesting in the Cosmos trial, again, as I had shown, is that there was a significant 38% reduction in lung cancer in Cosmos for those on a multivitamin versus placebo. That was one of the secondary outcomes. Incidentally, in the Physicians Health Study, there wasn't a significant reduction. I believe it was a 16% reduction in the Physicians Health Study 2. We didn't see that here for at least based on a metabolomic risk score. But there's lots of ways we can start to delve a little bit more carefully into some of these mechanistic pathways, understanding what is or isn't happening. So I'd like to pivot a little bit from the biomarker-based outcomes as part of a hybrid trial design in Cosmos to some other clinical outcomes. And what I'd like to present is just a series of findings that we've seen in Cosmos that have highlighted the role of the multivitamin on improvements in cognitive function across a few different sub-studies that are nested within the Cosmos trial. So I'd like to start with a project that we called Cosmos Mind. This was looking at more than 2,000 Cosmos participants that completed telephone-based cognitive assessments at baseline, 1, 2, and 3 years follow-up. And one thing that was interesting just to show is just the overall effect. So we found that those who were taking the multivitamin daily versus placebo had significant improvements in cognition over time. Now, one thing you'll also notice in this chart, because the active group in blue is going up, improving cognition, well, wait a second. So is the placebo group. Obviously, we have to think carefully about training effects. Once you take a cognitive assessment the first time, there's going to be a learning effect there. When you go through it the second time, you might know what things you might be expected to memorize, or object recognition, or other types of testing that takes place. That being said, even when you account for that, the multivitamin still emerged as having a significant effect on the global cognitive function score. One way to translate this when we think about these findings is to first on the left consider just at baseline, we have the x-axis for age at randomization, and then we look at the baseline global cognitive composite score. Again, this is summarizing many different types of telephone-based cognitive assessments, and that's on the y-axis. Excuse me, sorry about that. And you can see that there's a negative slope. Shockingly, unfortunately, as we get older, our global cognitive scores are going to lessen. And we can take that slope as a starting point and comparing that against the treatment effect and make a conclusion that daily multivitamin supplementation appears to slow cognitive aging by about 60% or about 1.8 years over 3 years of follow-up in Cosmos. Now, whether this is a real metric, whether it's an overstatement, perhaps an understatement, is certainly a matter of debate. Other ways that this can be looked at, and I don't have this finding to show, is that the Cosmos Mind group at Wake Forest also had looked at mild cognitive impairment and dementia as outcomes on their own. But because the study was only in about 2,200 participants at baseline, we didn't quite have the numbers to be sufficiently powered, but some of those hazard ratios were still trending in the right direction, which raises important questions about not just the short, but the longer term impact of taking a daily multivitamin on mild cognitive impairment, dementia, and other brain-related health outcomes that actually Cosmos is very well positioned to look at. Now, the best way for any study is to have replication, maybe not direct replication, but still replication. And in fact, in our Cosmos Web study, this was in conjunction with our collaborators at Columbia University, we had enrolled more than 3,500 Cosmos participants, again, with assessments of a web-based series of tests that were assessing episodic memory. These were done at baseline, 1 year, 2 years, 3 years follow-up. Once again, the multivitamin, those taking the multivitamin daily versus placebo had significant improvements in both this MOD-RAY test for immediate recall, and also looking at it not just at year 1 for 1 year change, but also across all 3 years. In fact, we also had some very interesting signals that had emerged for the cocoa extract or cocoa flavanol intervention here too, that are also warranting additional work and delving into it too. And again, if we use that same cross-sectional analysis approach, as we did for the Cosmos Mind study for the telephone-based assessments, we saw once again that just based on that slope at baseline by age for how they scored, that the effect of the multivitamin intervention had improved memory performance beyond that by placebo by the equivalent of about 3 years of age-related memory change. So it was effectively countering, the multivitamin was countering the changes in the immediate recall scores as part of this MOD-RAY test over the 3 years of the Cosmos intervention. Now, does this mean it's reversing how we our cognition? Maybe not reversing, but maybe serving as resistance to the power of age, chronological age on cognition. Finally, we were able to also have our clinic subcohort. Now, I haven't been talking too much about our clinic subcohort, but we also had, as part of our in-person assessments, a series of what we would consider to be more gold standard assessments of cognition, both at baseline and 2 years follow-up. And here, once again, we had seen that for our global cognition composite outcome, it was not quite statistically significant, but there were still at least positive improvements for our primary outcome. But more to the point, there were improvements in the secondary outcome of episodic memory, looking from baseline to 2 years in terms of the change. And so we once again saw that in this last part of the green box below, that if we take these changes that we're seeing in episodic memory, that these are translating to, again, fairly substantial changes or improvements in aging or less aging in episodic memory versus placebo. So for us, we have a telephone-based assessment, we have an app-based or computer-based assessment, and then we have an in-person, more kind of standard clinical assessment for cognition, all showing parallel findings here. And in fact, when we meta-analyzed these 3 studies, now mind you, these are 3 studies within Cosmos. So we tried to focus on those that were not necessarily, they were in each of in one of the studies alone and not necessarily accounting for, because there was some overlap here, but not a lot of overlap between the different sub-studies. But more importantly, what we had seen is for both global cognition as well as for episodic memory, for both of these, there were substantial improvements in cognition. Overall, pooling these data together for those on the multivitamin intervention versus placebo. And as part of our findings in this paper that was published last year, we basically found that it was equivalent to the average to the difference observed between participants 2 years apart in age. In other words, by taking the multivitamin over this period of time, you had 2 years less cognitive aging. Now, what's interesting is that there's questions about, well, what's driving this change if it's real? Obviously, we want to continue to think about how we reproduce it. But this is the beauty of a hybrid trial. When you're testing your primary outcomes and you're layering it with biospecimens, other types of data collection devices, thinking about wearables and other things too, and then combining that with these different outcomes such as cognition, we went back and looked in the epigenetics study, as I had shown you from before. And now when we look at it from the context of who was in both the epigenetics project as well as were included at least in the Cosmos Mind study, to see whether improvements in the clocks might start to explain the effect of a daily multivitamin on cognition. So by conducting some analyses, again, our data are somewhat limited. We're looking to hopefully expand upon this in different ways to understand more, but we do see some potential evidence supporting a role for epigenetics as mediating the association between taking a daily multivitamin and improvements in cognition. Again, we need to expand out the samples for this, the sample size for this to really understand this in more detail and improve our statistical power, but still insightful. Just a few more just brief comments on a few other projects that have been published or others ongoing or at least under review. Again, the advantage of a trial like this in Cosmos is that we have many other outcomes that we're collecting beyond those I've presented, including the primary outcomes. We've looked at the effect of the daily multivitamin and incident self-reported fracture. One thing just anecdotally that I was underappreciative of in an older population is just the frequency of fracture over time, even in what we thought were generally healthy adults, older adults at the start. But unfortunately, a multivitamin is not a panacea for that. We did see some suggestions for pelvic fracture, but even with the nutritional content in the multivitamin, the typical multivitamin, Centrum Silver multivitamin, we saw no effect for a multivitamin on fracture in Cosmos. Furthermore, we're looking at physical functions. This is tapping into some of the in-person assessments, and also we are expanding this to some of the self-reported measures of functional status in the main Cosmos trial. We're not seeing a lot of evidence supporting a role for a daily multivitamin on physical function. This is currently under review. But that being said, we still have the opportunity to start combining this with some of the other things that we've measured to understand whether there are biomarkers that could be explaining or not explaining some of these effects. Just one note, when we were looking at grip strength, we did find that there was at least some systematic error that had happened with our assessment tools. But that being said, overall, we were not seeing very strong effects. Furthermore, this is also work under review right now. One of the interesting things about the Cosmos trial, and one of the advantages perhaps of doing a remote-based clinical trial, which is what Cosmos consisted of, we of course did have a clinical component, a smaller in-person component, but Cosmos ended at the end of 2020, which, as many of you are aware, coincided with the start of our lovely pandemic. And through the efforts of our collaborators, our investigators, as well as our participants, we were able to continue the trial through the end of 2020. And so as a consequence of that, one of the benefits is that we can at least start to look at the role of a daily multivitamin on the risk of developing COVID, because we caught those outcomes as those had initially emerged. And so what we have seen is that at least in intention to treat analyses, so based on how you were initially randomized to Cosmos. Granted, that was about 2 to 3 years or so before the pandemic, but at least in terms of intention to treat analyses, we saw that there was no overall significant effect on total incidence of COVID-19 as self-reported. If we look at a secondary outcome of hospitalization or death due to COVID, there's a little bit more of a signal emerging, but we just didn't quite have the power. Back to the question of per protocol analyses, again, something that's always good to be considered. We didn't see any real big changes in terms of our outcomes. But what's interesting is that when we did start to look at the effect of a daily multivitamin in Cosmos on COVID-19 symptoms among those that were diagnosed with COVID, because we didn't just get COVID outcomes, but also number of symptoms. There we did start to see some interesting signals about perhaps the role of a daily multivitamin, maybe not in terms of preventing COVID-19, and we're curious to look at this in the context of other types of infections, but also in terms of the symptomology, there might be more evidence here suggesting not just an intention to treat analyses here with those with more than 3 symptoms, but also, excuse me, for those that in the per protocol analyses, there was a pretty consistent signal that seems to be emerging about perhaps improving the number of symptoms that you might have when infected. So it gets into the management of it per se. There's many other studies that are ongoing, hopefully that will be published in the months and years to come from Cosmos that go beyond what you've seen here. So to wrap up, what do we see beyond the Cosmos when I think about hybrid clinical trials in particular? It's important to reemphasize that when we think about nutritional and dietary recommendations, that's still the optimal approach to health, not just on a public health basis, but also individual health basis. We start with what we eat. Dietary supplements, though, are here to stay. I think there are many naysayers out there that would argue it's tossing money aside, tossing it out the door. That might be true. We just need more research and studies and trials to really understand that. And I think this is where I think very well-designed hybrid clinical trials that are truly cross-collaborative can really take dietary supplement research and make it much more meaningful and also informative for the scientific community as well as for the public. First and foremost, clinical trial design matters. I think it's often an underappreciated component, but by designing a hybrid trial from the start with these questions and considerations in mind, no matter what the size of the trial is or the length of the trial, the outcome is, thinking this way will enrich the science that comes out of it. Primary outcomes don't need to be major morbidity and mortality. They can be healthy aging outcomes across the lifespan. But it's incredibly important to incorporate the biomarkers and intermediate outcomes of interest. Ultimately, we want to try and create cohesive evidence so we can make more informative and broader-based dietary supplement guidelines. And understanding that dietary supplements do still have important roles within the framework of not just whole person health, but also precision nutrition and medicine. So with that in mind, thank you for your attention. I look forward to your questions and providing some context. Thank you.
Thank you very much for walking us through your work with cocoa, multivitamins, and their effect on health outcomes. So we have multiple questions in the chat. People are really excited about your work. I'll also, if anyone wants to actually ask their question, please raise your hand and I'll get you unmuted. So I'm just going to take this first question. How has the increasing costs impacted the formulation of cocoa?
That's a good question. I honestly don't track the prices of cocoa supplement extracts. There aren't that many in the market, honestly. There are a few products that are out there. I don't know if they're really in stores or if they're online. They're not as heavily marketed as all the other supplements that are out there. But I would imagine it just intuitively, if prices of cocoa, whether through just changes in our climate or changes in taxes or otherwise, if the price is going to go up, I would think that in turn the supplements themselves would go up in price too. But I honestly don't know the data on that.
Great, thank you. Can we unmute Barbara Sorkin?
I think I'm unmuted. Can you hear me?
Yes. Hi, Barbara.
Hi, Howard. Thank you. That was really I love the way you guys structured these trials, the Cosmos trials, with the biomarkers built in so that you can understand some of what's going on under whatever outcomes you actually see. I learned that from the Big Ginkgo trial. You build in outcomes that are going to give you information, even if you don't see an effect of the dietary supplement. So I had two questions. One, the big chart that you showed, showing many different outcomes, there were a few that seemed to trend actually worse that was for the multivitamin mineral, I think, including Parkinson's disease. You mentioned the fractures. One that I thought was really strange was that it looked like there was maybe a trend towards increasing rectal cancer, although colon cancer seemed to be decreased.
Right.
So maybe
Yeah.
the other question I had was many different outcomes. I think this is relevant to this. Could you talk a little bit about how you corrected for that in the statistical analyses for significance?
Yeah. So first, with regard to the multiple outcomes, that was the slide that was for the metabolomics work that we had looked at in Cosmos, and we had some signals in different directions. You're exactly right. It's a little unclear still. One of the challenges with a lot of the biological aging scores or the different outcomes that come out of it is understanding exactly what the clinical relevance is. Some of these scores that are generated have been linked with, say, colorectal cancer or rectal cancer or some of these different outcomes that are going up and down. But the one piece that's massively missing from the trial landscape, and even we're constrained to some extent in Cosmos, although with time we want to look at this more carefully, is that ideally you want to look at the effect of a dietary supplement, whether a multivitamin, cocoa, or otherwise. You would love to be able to look at the dietary supplement, the intervention, the mechanism, and how much that is mediating whatever that actual clinical outcome is. So ideally, if we're looking at, say, colon cancer, we would look at the randomized intervention, the changes in the metabolites that reflect the colon cancer score going up or down for that matter, and then who actually got colon cancer and who didn't. Those are the much more definitive types of studies that we need to think through, and that could be done not necessarily as one humongous trial, but rather some thoughtful nested case control case cohort designs that can help to address that. To your second question, Barbara, we typically don't often correct for multiple testing. And we do that for two reasons. One is that we want to let the results stand on its own, and we also try to take perhaps an overly conservative stance on how we interpret it. At the same time, with a lot of the trials that are done in general, not just Cosmos or Vital or other trials that have been conducted in dietary supplements, they're meant to be as much kind of hypothesis generating. And as one case in point, one finding that came out of Cosmos was that the cocoa extract supplement was linked with self-reported improvements in migraine. And that actually turned into a separate, smaller trial that a colleague of ours, Dr. Pamela Riss, is now undergoing called Coral to look at shorter term effects to see if that can be repeated or reproduced in different ways. So should we correct for multiple testing? Of course we should. If we do, we do it. If we don't, we try and be as conservative as we can. We don't want to overstate our findings. But at the same time, these types of trials have a lot to offer as well.
Thank you so much. And I'm wondering about, microbiome changes on the multivitamin mineral supplement, whether you see any of that, and do you have any data on exercise?
Yeah, so for the microbiome, we did see some changes, at least more functional changes in some of the components of the microbiome. For the multivitamin intervention, we also saw that for the cocoa extract intervention. One of the challenges that I personally have is that I think the microbiome is a fascinating area, but understanding, first of all, distinguishing between kind of the day-to-day variation in the microbiome versus a change in the microbiome that's truly due to a dietary intervention, whether a supplement or otherwise, and then understanding what those changes mean in the context of disease outcomes is something that still is an important research gap that we need to figure out how to best fill. And maybe those are more mechanistic studies in that regard.
ODS just funded a center that's looking at that specifically for blueberries. What organisms,
Fantastic.
what enzymes change and what exactly they seem to do in terms of metabolites produced and their cardiovascular effects.
Yeah. So it's really important to try to entangle that. And that's where the hybrid design for a trial really becomes important, because that really is what empowers one to be able to look at that more carefully.
Thanks so much.
Um, so how was product quality control factored into the meta-analysis? Did the studies that reported a positive effect use supplements that were third-party verified?
Yeah, so in the context of the Cosmos trial findings on their own, we had done actually quite a bit of work to ensure that the quality of both supplements were intact throughout the trial. So stability testing, different conditions, and also these were two products that had a history to them, so it made it a little bit easier for us to trust their stability and safety profiles going in. Um, for the meta-analyses, for example, that from the U.S. Preventive Services Task Force, when they're defining a multivitamin, it's a little tricky because they're not all the same thing. You've got different levels of nutrients contained in those different multivitamins, different types of, it's classic heterogeneity. But it does raise an important point about dietary supplement research, which is that whether for an individual trial or combining trials together in some pooled analysis or meta-analysis, trying to do a good job more collectively about standardizing the interventions across the board goes a long way to ensure that the findings that come out of that work are going to be more scientifically valid. Oops, Jasmine, I think you're on mute.
Yeah, sorry. I'm going to have one last question. If I didn't get to your questions, I'm going to compile these and send them to Dr. Sesso. So how can researchers strike a balance between leveraging industry resources and maintaining scientific independence?
That's a fantastic question, and it's a really important question. I think, especially as it pertains to dietary supplement research. Um, on one hand, we want everything to be fully independent. The work that I've been involved in over the years has always been investigator initiated. Regardless of whether it's investigated or initiated or not, I honestly feel, in my experience over more than 2 decades of research in dietary supplements now, not just in big trials, but in smaller ones, you really can't live without each other. A lot of times if you're testing a supplement, ideally it needs to be supplied by somebody. Who's going to supply it? What's that arrangement? So it would be great to have some best practices, as you've indicated in the chat for your question. Because I think ultimately, at the end of the day, we want to make sure that there's trust in the findings that are presented. Trust not just in the scientific community, but also in the broader public health segment, in the broader country, right, or world. Because a lot of the trials that we all do collectively have important implications. Not enough trials are being done given the regulatory framework that we're in with DSHEA. We don't have the same thresholds at this point that are required for dietary supplements in the same way that we might for a prescription medication. So with that in mind, we need the science to be solid and trusted so that when we have our results, whether they're in agreement or not with what we think, that we trust the data and that the process is clear and transparent as well.
Okay, so I want to give a big thank you to you, Dr. Sesso, for taking your time out of your busy schedule, giving that exceptional presentation, and just kind of gracing us with so much valuable information. And I want to also thank everyone that joined today. Your engagement makes sessions like this impactful. I've seen so many questions in the chat that, again, I will compile them and send them over to Dr. Sesso. I hope to see you all at a next seminar.
Thank you.